Hello, everyone. We're just going to be starting here in a few more minutes. Elaine, are you there?
Yes, I'm here.
All right, perfect. All right, well, I guess we can get started. We're just going to be starting here in a few minutes. Okay, yeah, so I think that we're ready.
Okay. Welcome, everyone. We're going to get started today. This is the orientation for data science for biomedical data. So for some of you that have already participated in the orientation that happened last week, this introduction will be a little bit of review for you. But for those of you that were not able to come to that orientation, this will be fresh information. So first off, I want to thank LSU Biomed as well as LBRN for funding these training modules available. Biomed is under LSU. It's a group of cores, and it stands for the Division of Biotechnology and Molecular Medicine. That's the Biomed down here. Gene Lab is one of the cores within Biomed, and I'll talk a little bit more about that at the end of the lecture here. And then LBRN is Louisiana Biomedical Research Network, which is a consortium of universities that work together to further Louisiana's research potential as through collaboration and communication. But that aside, let's go ahead and get started as to the information that you guys are here for today. So a little bit of overview about what these training modules are. So these training modules are designed to be completely asynchronous, and that is because many of us have very busy schedules, right? So the opportunities available here for you to jump in and jump out around your schedule and work with on your timelines really should benefit you here. Additionally, you're not in this alone. So the purpose of these programs is to help guide you through the information and provide you with interactions similar to how you would with, say, a professor or an expert in the field that would be teaching a similar course. So for these training modules, we'll have access to a variety of mentors that will interact with you both through lectures, as well as be accessible through online chat, which has a variety of options for you to reach out to them, and email, of course. And once you have gone through the program, once you've reached the completion of the program, you will receive a certificate so that you can prove that you completed this training module and have a better understanding of data science. Now, depending on what institution you come from, that certificate will look a little different. So if you come from LSU, your certificate will be customized to LSU and be signed by Dr. Gus Kasoulis. If you come from LBRN, your certificate will also be customized, but it will always be signed by the LBRN's bioinformatics core lead, Chris Taylor. If you are not part of LSU or LBRN, your certificate will be similar to a standardized certificate that you would receive if you completed the program independently. So that being said, there are three main things that you're going to utilize while you go through these programs that are going to help you through the lesson plans or you'll interact with frequently. The biggest one you're going to interact with frequently is the OmicsLogic portal. I'm going to show you how to register for that in a little bit. The OmicsLogic portal is essentially the location or the website you're going to go to and access all of your lesson plans, where you're going to see your syllabus for the program, as well as access a variety of mentor interaction options. The next thing that you're going to interact with fairly frequently is the TBio platform. So the TBio platform is designed to allow you to input bioinformatics acquired data and then run it through a various pipeline of analyses so that you can go through the normal process that a bioinformatician would to analyze the different data, except this is a much easier for those that don't have the expertise to perform the analyses through their own coding. However, you're not just going to be using this platform, you are going to learn how to code to some extent. This program in particular is going to involve much more coding than the other two that are being offered this semester. So you're going to be doing this partially because you guys are a coding course. You will have some work with the coding playground. And so the coding playground is essentially a virtual or online playground or a coding environment so that you can practice your coding online without having to establish a coding environment on your computer. Now, because you guys are a coding based training module, you'll likely also have to set up a coding environment on your own computer, but this, as you learn, will be available to you so you don't have to go through that process right away. So this is an intermediate based difficulty course. This basically means that it's targeted towards individuals that have an understanding of both biological and data science concepts, as well as some experience with statistics and some experience with coding. You don't have to be experts in any of these things, but the expectation is that you are able to understand some of the information given to you from the get-go. So there are 2 other difficulty level offered training programs this semester, which we'll be talking about transcriptomics tomorrow if you're still interested. And we were talking about introduction to bioinformatics yesterday, and that recording will be available online. So what exactly is this training module? So this training module, the orientation is obviously today, but you have access available to you already if you've registered. This training module is designed to help you analyze bioinformatic data. That's from everything from data wrangling all the way through visualizing your data. Many of us have seen visualized bioinformatic data if you work in biomedical field, but this course is designed, this training module is designed to help you go through the actual steps and understand the analyses that are going on behind the data visualization, because there's a lot of steps to get to that visualization point. Some of the concepts you're going to cover is statistical concepts and their applications to biological data, machine learning and deep learning, Python and R coding, as well as coding challenges so that you can start to understand how to do the coding yourself, as well as how data sharing. Now, you're also going to have a variety of ways to learn this. There's hands-on exercises, there's tutorials, there's lessons, there's the mentor guided lectures. There's plenty of learning opportunities here that will help lead you through this process. And it's not just going to be conceptual applications. This training module is targeted to also be able to practically apply this information in your research world, in your clinical world, and the variety of other applications that data science finds itself in. And like I said, you're going to be able to practice this as you go, so you're not going to be stuck just learning the concepts. So I'm not going to go through all the details of the lecture or of the syllabus, but these are, here's your syllabus. These are the finalized dates. Later on, Ilya will go into more detail about that. Additionally, there are other resources available to you, especially if you're coming from LSU or LBRN. Like I mentioned at the very beginning, there's LSU Gene Lab, and LSU Gene Lab is available to everyone within the LSU network or the LBRN network. Basically, what LSU Gene Lab is designed for is to provide consulting, as well as a variety of experimental services, which range from next generation sequencing all the way to automated cell counting. I listed a few of those services here. This is not their full list, but this laboratory and this service laboratory is available to anyone within those networks and can very much help your research if you're not familiar with one of these techniques. Additionally, LBRN, like I mentioned, Chris Taylor is associated here with the Bioinformatics, Biostatistics, and Computational Biology core. This core is basically designed to provide consulting, mentorship, and training focused in bioinformatics and data science fields and how they're applied to biological data. Now, this is targeted towards faculty and PI, so keep that in mind if you reach out to them. This program is also designed to create a project at the end. Now, I did want to give this information to individuals that every year LBRN holds a conference focused on computational biology and bioinformatics. This is usually around April or springtime. So once you complete the project in this training module, you'll have the opportunity to present that project at this particular conference or any conference of your choosing if you're interested in that. Additionally, some new information I received today is that there is also a master's program focused on bioinformatics available at the LSU Health Center in New Orleans. So if you're interested, you can look that up yourself if you want to continue your knowledge into the bioinformatics field. And finally, if you're a faculty member or a PI at another institution and you're interested in having these programs or training models offered at your institution, you can contact Ilya Brodsky, who is the CEO of Pine Biotech, who is offering these training models at Ilya@PineBio for more information. So I'm going to hold here before I show you how to register, but I'll take any questions if anyone has any questions about this process itself. If not, we're going to move on to the registration.
No, sir.
Okay, so if we don't have any questions, I'm going to go ahead and switch screens here. I'm going to show you how to register on the OmicsLogic portal. So for those of you that have not done this already, if you're interested in this training module, you have to register on OmicsLogic before we can add you to the course, the training module. So to do so, you're going to want to go to learn.omixlogic.com. When you come to this page, you will be able to see clearly in the front how to register for free. And they put that link in the chat. I will also mention before I go through this that there is another website. I'll put the link in the chat that will go through all the other training programs and training modules that are offered at LSU or through the LSU Biomed and LBRN network this semester. So you can go to that link if you're interested in the other training modules. But like I said, go to learn.omixlogic.com to register for any of these training modules. The way to do so is to click register for free down here. And then once it loads up, you'll see on the right-hand side a sign in or sign up section. All you have to do once you get to this section, if you already have an account, you can sign in here. If you don't already have an account, there'll be a small button you'll have to click to expand the information to allow you to register for learn.omixlogic.com. Great, so here. So if you already have an account, you'll sign in here. If you don't, click on create an account right here. This box will expand for you and give you and ask you a little bit more information like your name, the institution that you're with, et cetera, et cetera. But since I already have an account, I'm going to go ahead and log in so I can show you a brief introduction of the profile on OmicsLogic and how to access the information that you'll need to access for this training module. So when you log in, you'll open up to this page. I recommend going to your profile by clicking on right here, welcome back. Your email will be listed. And it'll take you to your profile. Once you're added to the training module, you'll have two ways to access the syllabus easily from your profile. The first one would be come down here and click on the data science badge. The other way to do so would be go over here to the programs tab at the top of your profile page. Click on that. And once this loads, you're going to go to Biomed Data Science for biomedical data. Before I jump there, I do want to show you how to access some other things here on your profile page. One that you'll use frequently is your courses. So once you have started an asynchronous course, you're going to be able to find it under the course tabs to continue it. Because again, this is on your schedule, and then you might not always be able to complete a lesson in one sitting. But what you can do is come to the courses tab and find whatever courses you have started and easily access them later on. You'll also be able to access any certificates you receive, and I can show you what one of these looks like now. Once this loads. So this is what your certificate of completion will look like. And like I said, depending on the institution you come from, it'll be more customized and it'll be signed by a different individual. But if you're coming from an outside institution, it will look very similar to this, and it'll be signed by an individual associated with Pine Biotech. So if I go back here, I'm going to show you the syllabus page very briefly. Under the programs tab, like I said, you can go to your syllabus at any time, or the program page where you'll find your syllabus. So here's your program page. If you have not registered already, you will not have this pop up here. This is because I've already been registered. If you haven't already registered, if you go to this page, this link, you'll be able to scroll down and you'll be able to find pricing in this area about how to access the program. But since most of you should already be registered, you'll be able to scroll down and you'll find today's the first lesson. So here's your find the syllabus information and also the information for each individual lesson. Today being October 7th, you can see this is the topics that are going to be covered today. And here are the associated resources that you should go through after this lesson. Now, once you complete everything with this lesson, you'll be able to click complete lesson here. And once you have done that for every single lesson in the program, you'll receive your certificate. Okay, so that being said, that's all I'm going to cover today. I'm going to go ahead and hand off to Ilya with Pine Biotech, and he'll go over some more details about the training program.
All right, thank you very much, Lane, and welcome everyone. Good to see that everyone has joined. And I'm going to start sharing my screen. So what I want to do in my part of the presentation is go over a little bit what we will cover in each one of these parts. So already Lane mentioned that we will be talking about data science, we'll be talking about machine learning, and we'll be speaking a lot more in detail about every aspect of these things. Now, the major objective of this data science program and how it is different from some of the other programs that we've been talking about yesterday and we'll talk about tomorrow is that this particular program is designed to give you some more general skills in addition to your ability to analyze biomedical data. So what do I mean by that? First of all, data science in itself is very similar to bioinformatics. So you could define bioinformatics as the intersection of computational tools and methods to organize and manage your data with a focus on biology, whereas data science is a lot more general. So it could be applied to a lot more different applications besides biology itself. And so you can kind of say that bioinformatics combines a lot of the same elements, but with this unique focus on biology. At the same time, when people hear the word bioinformatics, they tend to think of very specific applications of bioinformatics. So people would think about BLAST and kind of sequence alignment and things like that. However, I want to talk today about how the nature of sequencing data is changing, and a lot of the ways that we analyze this data is also changing, bringing to the forefront this need to have data wrangling, data analysis, data visualization, and machine learning skills in what we are able to do. And so I'll talk a little bit more about what we will be able to do in the context of this particular program to help you kind of get those skills. So the program will combine three main modules besides some introductory material. These are the three main modules that you will have to complete: BioML-Logic, BioML-R, and BioML-Python. Now, BioML simply stands for bio data, and ML stands for machine learning. But essentially, it will cover statistical analysis and machine learning that starts from data preparation for statistical analysis all the way to interpretation of what you have been able to identify using a variety of analytical methods. So besides these three modules, there are some additional modules that you can choose to complete. They're not a requirement. So for those of you that have less background in biology, we recommend the course called Bytes and Molecules. Kind of talks a lot about how to apply different types of methods in bioinformatics to different domains, like let's say medical or maybe basic biology. It also introduces a lot of the terminology that you would have to know to orient yourself. So if you come from more of an engineering background, maybe you're not so sure what's the difference between a gene and a mutation, exon and intron, or maybe sequencing DNA as opposed to RNA. And what does that mean in terms of data? Because a lot of the words are used in a different way. So that's something that you can clarify in those introductory modules. And then for some of you that do not have any background in engineering, so you have not done any coding at all, there are some introductory modules like Introduction to Bioinformatics in R and Introduction to Bioinformatics in Python. So those would be useful for anyone that comes without any coding experience. What these modules provide are some exercises that clarify what is the problem or why are we doing what we're doing, how to address this problem from a coding perspective, how to use existing code and maybe modify some of the settings and outputs, and then apply them in an effective way to a data. So here you can see an example from getting started with bioinformatics in R, how to load data, how to visualize it, maybe how to read different types of data, maybe tables or sequences, and how to apply some of these commonly used bioinformatics tools. So this is just to get you started. When you move on to the more advanced courses like BioML in Python, for example, you can see that they will use project examples with curated data sets taken from publications. So in other words, a lot of the methods will try to replicate the analysis done in that publication, having to code every step of the way to be able to understand what is the method, what are some of the assumptions about the data, and how can you choose between the different methods to really select the one that is most useful in the context of the research question posed by these different data sets. Now, besides that, we have this parallel. So the exact same thing that you will learn in R, you can learn in Python. Why R and Python? The primary reason is because R has a lot of commonly used packages. You will find typically outputs from different R packages in publications. A lot of the statistical analysis is very easy to use with kind of beautiful visualization right out of the box with minimal modification. So that's why R is very popular in bioinformatics. You have a whole a series of packages being developed every day to address new types of data and new types of analyses. At the same time, in Python, it seems like some of the things are going to be a little bit more challenging in terms of, let's say, visualization and making these beautiful figures that you can just take and use in your publication. But a lot of times, once you get into the syntax, Python tends to be something that people feel much more comfortable with after they've kind of overcome this initial stage of some technical things that you just have to practice and know. So these two languages are very popular, and we'll talk a lot about how to get started, how to set up your environment, and eventually how to use those in the context of a data set that you choose or you pick for your project. Just to kind of give you more in-depth information about how this is actually going to work. So here's an example from the Python version of the course. You can see that there are several different sections here. You essentially will be able to learn the code syntax just by reading about the code. It will explain every single piece of the code. What are we trying to do? Import packages, loading data, removing specific portions of the data, manipulating our data, and then you can practice. So essentially you can replicate this code, run it, and test your results, whether your results are correct or not. And so as you saw here in this little video, you essentially run that piece of code right in the browser, and it will tell you whether your result is correct. Besides that, you will then be able to take this code and modify it so that you understand how to apply it to different types of data sets. So each exercise is going to be first to understand the objectives and strategies, then to learn the syntax or how to code this in a particular language like Python, then how to test your understanding in these coding challenges. And so for the coding challenges, you will get a link to a Jupyter notebook that will typically have some initial code. And then once you look through that code, you'll be given an assignment how to complete it, how to maybe take something that you've learned in the lesson and apply it and write it out in code and make sure that that code works. So a Jupyter notebook looks like this. Essentially, it's like a Google document. In this case, this is a Google Colab, where you can take each portion of your code and run right in the browser. In this example, all of the code is already written for you, but in some cases, you will have to modify or maybe write full pieces of code yourself. And that way you'll learn how not to only learn about the syntax, how to also write it all out yourself. So let's talk a little bit about what the different sections are going to cover. And so this is an overview of all of the topics that we are going to be covering in this program. So we're going to divide it into 3 different parts. The first part is introduction. The second part is methodology, how to understand and apply these methods. And the last section is actually about the application to particular projects. And so the first session is going to give you a sense of omics data, terminology, and key concepts that you just have to know to get started. The second portion will tell you about the details of the methods. What's the difference between different clustering methods, or how to do dimensionality reduction using different techniques for different types of data, and what are the differences between statistical analysis methods and machine learning methods or deep learning methods. And then finally, you will get introduced to a number of different research publications with curated data sets, and you will see how this is used in clinical applications, in biomedical research, and in different aspects of what the industry is doing with clinical trials or with designing new drugs and things like that. So let's talk a little bit more about the different challenges that you should be able to address once you complete this full program. Typically, when people think of big data, they think of data that is intuitive and easy to understand. And in this case, especially in the world of biomedical research, typically that is not true. So there are several different challenges around it. First, this data is unintuitive. So you really have to have a lot of additional information to understand what the data describes. And number two, the data is not structured. So as you can imagine, a lot of these data sets that I have here on the screen come in their original form in an unstructured way. You cannot just take them and analyze them. So the first challenge that you should be able to do after this program is to structure data and prepare it for analysis. What kinds of data are we going to be dealing with? Primarily, we're going to be talking about omics data. This includes phenomics, genomics or DNA variation, epigenomics like bisulfite sequencing, so DNA methylation, or ChIP-seq, where you're looking at histone modifications, and transcriptomic data, including some other types of data sets like metagenomics. So all of these data sets have to be processed to be useful. And we'll briefly touch on how to process data, because essentially sequencing data looks like this. You have short reads. Those short reads have to be aligned to the reference genome. And only after they have been aligned to the right place on the genome, you can actually quantify how many you have and translate this into structured data that is quantified. So you actually have numbers associated with features like genes and samples that represent some biological conditions that we can compare and contrast. To do that, you essentially have to know some of the basic ways that some of these coding languages deal with data. And the way they deal with data, in this example, we're going to look at Python. So in Python, you have two major packages that allow you to work with matrices that essentially represent numeric data that you can manipulate and work with, and pandas that allows you to work with data frames. And so we will learn about how to take this kind of data, prepare it for analysis, load it into the environment that we're working in, for example, R or Python, and then that will make it ready to explore. And so once we've loaded the data, we will start looking at data properties. So how can we estimate different important parameters like distribution, like, for example, different ways of normalizing data to check that the data is kind of consistent with our assumptions about the data, and how to essentially prepare this data for the method that we will use in the analysis, because in a typical way, that method will have some expectations about the data that are important to make this analysis correct and make it interpretable. As a part of that, we will talk about different ways to visualize data. An important part of kind of playing around with the data is being able to, for example, extract different portions of your data frame, combine them together to find a data set that you need. For example, if you're talking about different types of conditions, and those conditions are defined by different sexes, different ages, different disease stages, you might want to look at just some of those. And so one way to do that is you have different types of categorical data, and you want to create a data frame so that you can analyze that, and that's called crosstabs. The other way is you can compare categorical groups. So for example, different age groups, different sexes, different disease types, and then you can create summary of continuous data, for example, gene expression, to visualize it and make it kind of statistically comparable between different groups. And finally, we'll talk about how to analyze and visualize continuous data. So if you have, for example, 2 genes and you have many samples that are defined by those genes, or in the other way around, you have many genes defined by 2 samples, how can you visualize the combination of this continuous data, for example, in a scatter plot? So we'll talk a lot about visualization. And so you will be able to take unstructured data, which could be gene expression, could be metagenomics, or even could be survey data, and essentially make sense of them and visually presentable. So here you can see a bar plot, a pie chart. So those are some simple tasks that you should be able to do after you complete this training. What essentially that gives you is some method of manipulating your data. So from random patterns, like you see here, you will get patterns that could be biologically interpretable, right? And you will be able to then differentiate between different types of signals to know that they are different, and to start thinking about how do you analyze many of these types of signals as you go into the analysis stage. So we talked here, spoke about data processing, essentially, preparing your data for analysis, and then starting to explore it. And so as you complete the full program, you should be able to understand the significance of experimental design, how the data was generated, to then how the data is typically processed, cleaned, prepared for analysis. Then what is the major trend in your data that you can use to define your statistical analysis and pick the right model, and then essentially use the analysis results to make interpretations as well as generalizations about whatever this data represents. So major kind of part of that is statistical analysis. And what we'll do in the statistical analysis section is we'll talk a little bit about different ways that you can make sure that your analysis has some significance. So what is that significance defined by? What are p-values? What are fold changes? What are those error bars that we typically see, right? So we'll talk a little bit about those concepts because they're extremely important for us to understand before we jump into these more complicated methods of analysis like machine learning. So you will be able to, for example, use packages that can simplify your comparison between multiple types of data and make sure that you understand different parameters like level of correlation, or like I said, p-values and things like that. You'll be able to extract different elements like genes based on statistical properties of an analysis. You'll be able to understand how to use those different statistical parameters correctly. You will be able to also test and validate that what you are finding is consistent with biological literature or research literature out there. You will also be able to summarize your data in intuitive and useful visual format. So here we have a heat map, and you will have a whole way of going through the data to kind of summarize the significant part of that data based on your experimental design or the contrast that you've built into your analysis. We will also talk about different exercises that will combine elements of loading data, running loops, making sure that you know how to filter and save different components that you have in your data, maybe even print reports that is going to be very useful when you want to share your results with somebody else, visualize data, and make complex outputs. And we will do that both in R. As I mentioned, again, in R, you have very easy to use predefined packages, or Python, where you have to put a little bit more work into maybe sorting the data before you visualize it. And you also have to understand the different elements of the syntax. So we will go through that as well. Then we will come to kind of one of the major objectives of such programs. And one of the major objectives when you get to know your data closely is how to differentiate signal from noise. So I really like this picture here on top. You can see that even here, the signal is present, right? So you can clearly see that there's something there. There's kind of a snapshot of some difference, but the goal of the analysis is to really focus on the difference and distinguish them or separate the signal from the noise. And to do that in statistics, typically we can use a model for the noise with some assumption about the data. So for example, if the data was randomly generated, we can then assume that the noise is representative of that randomness. And we actually have a formula here for noise that kind of helps us explain or model this noise. And so we can start putting together the different elements that help us understand and focus on the signal. Machine learning, in many cases, uses the same principles, but it also relies on algorithms to take those differences between signal and noise and be able to apply them in more sophisticated ways. So that's why next we will turn to data mining and classification, and essentially go over some of the commonly used, what's called conventional machine learning approaches. These approaches have been kind of popular and used since the 1950s, really, so for a long time. In the last decade or so, they have become much more popular and much more easy to use for a number of different reasons, including complexity of data, the volume of data, and computational tools and technologies that we now have in our, made accessible to really be able to use them in a much simpler way. To really understand each one of these methods, we will also make different comparisons so that we understand the significance of different elements and different outputs of these methods, because in a lot of cases, people just apply them without a lot of understanding, without understanding of how to use these settings in some meaningful way. And so we'll go into the detail and we'll be able to really look at each method in depth, understand how they are related to each other, how to compare and contrast those different methods. What do they essentially mean? What are some of the algorithms behind these data? And how can we essentially explore our data and prepare it for analysis so that we know that we are using the right kinds of assumptions and the right kinds of methods, and they really make the analysis interpretable. So we will kind of review all of these methods in some detail to make sure that everybody understands also what goes on behind the scenes. We'll also introduce you to some standard ways of implementing this using such packages like scikit or scikit. So how scientific methods are used in Python, including different packages that are commonly used for this in, for example, in the R language. We'll also try to address some of the linear algebra kind of elements of machine learning. So here you have singular value decomposition for PCA. So we'll try to make sure that in some layman terms, everybody understands where this kind of analysis comes from. We'll talk about how this is actually useful to understand where this comes from, because now you will understand the significance of different properties of these objects that are produced by Python and R packages, and you can actually get a lot more information out of that. And you will be able to compare and contrast different methods for optimization and for really making them less overfit and more generalizable. Now, in many cases, like I mentioned, these methods include some algorithmic understanding. And so we will talk briefly about some of the algorithmic components here as well. We'll talk about feature selection as an example of a greedy algorithm. We'll kind of give simple illustrations of how you can understand these different algorithms and how they rely on some of the statistical properties of your data, as well as the different types of assumptions that this algorithm will have, so that you know what is the meaning of the different settings when you try to run these analyses, and also understand the outputs and where they came from. So what is the reason why you're seeing what you're seeing in the output of these particular data sets? We'll also talk about different applications of these methods to a variety of different types of biomedical omics data. So we'll talk about it in the context of diagnostics and cancer biology. We'll talk a little bit about single cell and tissue composition, as well as microbiome and infection, or how we can use this proportionate data to infer some meaning about different conditions in terms of diversity, complexity, and things like that. So I think a lot of exciting applications of some of these data science methods. Now, as a result, you will also be able to read data and understand what each data set represents. You will be able to distinguish between different methods that are typically mentioned in publications. So here you can see one of the publications that we will use. It has all of these terms: support vector machines, random forests, different molecular profiles. So you'll be able to kind of read some of these methods and understand what they mean and actually know how to implement these methods. And as I mentioned, we will provide different curated examples within the course that you can then choose to expand on. Essentially, all of these curated data sets are provided as a standalone module. You can go through it and understand how the authors performed the analysis and what does the analysis really mean? What did they actually publish on? So to summarize, the program is going to be separated into 3 different parts. The first part is going to give you some introductory materials so that you understand the terminology and key concepts that you will need to be successful in this module. The second part will focus on different methods and in-depth understanding of the methods, parameters and settings, and kind of the logic behind those methods. And then finally, you will be introduced to a number of different projects that you can use to, first of all, understand how these methods have been used by experts that wrote the publication, and then potentially choose to develop a project of your own using some of these methods that you have learned how to code. And essentially, the objective of having a lot of these exercises done in a Jupyter notebook is also to provide you with a whole collection of different scripts that you understand, that you can modify, that you can read, and then have little differences in data sets that you know how to adjust the code for, so that you can essentially use this for your own project. So at this point, I want to ask if anyone has any questions that I can answer. So if you do have any questions, please put them into the chat box. If there are questions that have been posted already and I have not seen, maybe you can help me, Lane or Jesvi, see what I need to answer. Okay, so if there are no other questions, okay, 1 second. What is the best language? Okay, so it's a good question. What is the best language? Python and R. I don't know if there's such a thing as the best one. So there's a funny kind of answer that I read about this. So has anyone here heard about Visual Basic? Like in Excel, you can write VBA scripts. Yeah. So, that came around a long time ago, right? Visual Basic was since the beginning of Excel. It's been around, and many people complained about Visual Basic. It's a very difficult kind of thing to learn. And then once you learn, a lot of things don't work, right? So people have been talking about this Visual Basic language disappearing. And the fact is that today, many people still use Visual Basic and use it effectively if they have learned and kind of applied it in an effective way. I think in a similar way, it's difficult to say that, one language supersedes completely another language and everybody just goes and learns a new language and that becomes the best, the most popular, the most efficient. I think what we are seeing is that a lot of people use these languages for different purposes. For example, a lot of people in statistics use R and are encouraged to use R, particularly because there are so many different easy to use packages. And so if we see bioinformatics from that perspective as a way to analyze our data, explore our data, I think R is a very useful way to get started. I think it's intuitive, depending on who you talk to. Some people like it, some people don't like it, but it's a good, well adopted and kind of bioinformatically validated language. Python, I think, is easier in terms of syntax. It has less, I think writing in Python, once you understand kind of the logic behind it, becomes easier, but it's more work. So it's not just like modify a couple of pieces of how you call some function and it just works. You have to start doing loops and you have to filter and you have to call different packages. So a lot of this, I think, tends to be a little bit more complicated in Python. But Python seems to be much more adopted in other fields. For example, besides bioinformatics, in other fields, it's a very useful language to know. Okay, so how can you start? Again, like Lane mentioned, you can start by, first of all, taking a look at the full syllabus page, which is right here. And I'm going to paste this in the chat. And this link will kind of give you a good overview of everything that's and provide you with some links. So if you scroll down, we'll kind of talk about some of the major things that we will cover and give you a good overview of the full syllabus. Now, if you want to then participate in the program, you essentially can have two different ways. One, if you are already at LSU or LBRN, and then the best way is to do it through iLab. And we have a short video that we will send by email after this on how to do this on iLab if you are from LSU or LBRN. What's important to remember is that for LSU and LBRN, really, you can be a student, you could be faculty, you can also offer this to your students as well, if you think that they could benefit from such training. And the training, as already mentioned, is asynchronous, so you can essentially just use the whole semester to complete all of these modules and get your certificate. Now, once you're here, or let's say you're not from LBRN or LSU, what you need to do is you need to sign up on the portal. So let me ask you, how many of you have already signed up on the OmicsLogic portal? Maybe if you have signed up, put a number 1 in the chat box. Okay, great. Thank you, everyone. And Isa, I will address your question in just a second. Okay, so if you have not yet signed up on this portal is free, and I'll just show you a little bit how you can use this portal. So this portal is free, and it has everything that you need to get started and grow. So essentially from very basics, if you're completely new to bioinformatics, all the way to wanting to use bioinformatics in a more effective way to do research, you can do this through this portal. Once you're signed up, you can just simply go to the courses page. And on the courses page, you will have several different ways that you can navigate the material that you have here. You have tags here on the side. So for example, you can look at data science courses or machine learning courses, right? You have these collections. So for example, getting started or bioinformatics in R, bioinformatics in Python. So different categories of courses are organized right here. You can also look right here in the search bar. In the search bar, you will find a simple way to find the data. So for example, if I'm interested in cancer, you can see everything related to cancer. Or if I want to learn about DNA, so here you will see DNA highlighted, right? So that's something that you can do right here. If you just want to get started, there's an excellent collection of courses to get started, and you will find it right here, getting started. So if you click on getting started, if you're new to bioinformatics or you're new to omics and sequencing data, you can go right here. And you will see that there are several courses to kind of get into it. So Introduction to Bioinformatics is a very short course that you can complete for free. You don't need to pay, you don't need to register, you just complete this course for free. There's also a course called Bytes and Molecules. And this course is a little bit more comprehensive, and it has several different aspects that you can learn about, what is DNA? What are these sequences? How is sequencing data generated? What are proteins and protein structures? Introduction to genomics, transcriptomics, and we'll have probably by tomorrow, introduction to metagenomics. So you'll learn about different types of omics. If you want to just get started with coding, you can use this, Introduction to Bioinformatics Languages. So this is a course that's really designed to get you started. Now, after you've completed this, you will see that under your profile, you will start seeing points accumulating. And so when you see these accumulating points, essentially they're getting you from a beginner who doesn't know a lot to intermediate level to then an expert that gives you some validation that you know it. And you can also validate it by looking at the number of courses that you've completed and the certificates that you've gotten by completing those courses. Okay, so I see that somebody raised their hand. Oloyede, did you want to ask a question? And Filani, so go ahead, you can unmute yourself and ask your question, or if you would like to type it in, you can also type it. Yeah, go ahead.
Good evening.
Yes, good evening.
Yeah, good evening. Yeah, I want to ask, because I received the mail, because I remember registering for the, registering for this data science, omnic, genetic. So is it that the classes is going to be on Zoom or through or on the portal? We have to register on the portal first.
Yes, you have to register on the portal. So
Okay.
again, you go to the programs. Did you already create a profile?
I think I already did.
Okay,
Yes,
so
I
if you
already
can
did.
log in, after you log in, go to the programs tab. And right here, these are 3 programs offered by Biomed. So if you just type in here Biomed, okay, okay, Biomed is with 2 M's, Biomed. You can see the 3 programs that we're discussing in these orientation sessions are Introduction to Bioinformatics, Data Science for Biomedical Data, and Bulk and Single Cell Transcriptomics. So today we're talking about this program, Data Science for Biomedical Data. If you have not completed the registration, take a look at this link. It will look a little bit different from what I'm showing on the screen. Okay, thank you. Ojasvi just posted the link. So take a look there. And if you have any questions, then Ojasvi would be able to help you with that too.
Okay, thank you.
All right, great. And Filani, did you have a question?
Yeah, good evening, everyone. Yeah, my question is there a means for internship? Like, can somebody get interned in with you? Each person have the basic of bioinformatics already. Because I did the course in school already, so I have the basis of it. So I want to probably practice what I've learned or what I've known. Because I'm into data analysis and I've done data science already. But just for into practice. So that's what I'm asking.
Okay. Well, so yeah, there are different ways that you can use these different programs. One of them is to get training. And so if you've completed your training, then you should focus on your research project, right? So the goal of this portal is to train you and then give you the tools to work on an independent project. How do you do that? There are several ways that you can do it. If you already have a subscription on the portal or you're part of a program, you simply go to your profile. And once you are in your profile, you will see that if you have 10,000 points right here, or more than 10,000 points, you can navigate to the projects tab and start a new project. Okay, so what this new project will be is essentially like a publication. So you will see that all the different steps here will resemble a publication, abstract, methods, results, discussion, etc. And this will be useful for you to demonstrate that you have really learned a lot of these different types of data, different types of analysis, and can do biological interpretation. If you would like to have help for someone to actually work with you as a mentor, the way that you can do that is by joining the Research Fellowship. A Research Fellowship is a program that we run every semester. So right now we're in the fall semester. So we have the fall Research Fellowship program. It's going to go from September until the end of December. And essentially the goal is to prepare your project for the upcoming conferences in bioinformatics so that you can present. So if you need more details about that, you can actually reach out to our team. You can reach out to support at Pine Bio or marketing at Pine Bio, and they can guide you on how to register for the Research Fellowship. Isa, did you have a specific question that I can answer?
Yes, good evening, sir. I have a question on machine learning. I did a little project on it, like breast cancer prediction. But still my question is on already I give the question through the chat that DNA sequencing. I did a lot of things, but still I did not understand it. So please, can you help me with a specific website that I can go and register or I can follow so that I can understand the basic part of it? Thank you very much.
Okay, well, yeah, that's a good point. So the data essentially comes from the process of sequencing. And there are a couple of courses that I could recommend from here that you can actually get started with. For example, this course on transcriptomics will talk about how the sequencing, the process of next generation sequencing works. Or you can, this is for RNA. You will find the same for DNA. So you can look for genomics, for example, and here you will also have an explanation of sequencing and what essentially you can do with different sequences and how to analyze sequencing or process sequencing data. Depending on the level of complexity and kind of technical questions that you have, these are some of the courses that you can use to kind of get started and understand the process of sequencing, the different file formats, how to process them. And one of the major benefits of these courses is that they actually can provide you with an overview of how to build pipelines and use some data sets from our repository or from the public domain to actually practice how to do that. So here, for example, we have the T-Bioinfo platform. You will have access to this platform as a part of these courses as well. And what you will see is different ways to process RNA-seq data, so transcriptomic data, or genomic data. So here, mutation variants, you can see how to use FASTQ files or SRA files from NCBI and turn them into genomic variant calling files, VCF files. So these are some of the tools that you will be able to practice here. Inside each one of these pipelines, every method has a link, so you can actually understand how the methods are connected together. And then if you wish to go into more technical detail, you can follow those links. Okay, Kashaf is willing to work in the company. Great. So please send your CV to Ilya@PineBio as a bioinformatician data science. So always looking for great talent. So please go ahead and send us your CV. And all right, thank you. Ojasvi. Okay, are there any other questions that I can answer for now? Email, yes, so Ojasvi right there shared my email, ilya@pine.bio. Okay, so I think we are just approaching how can you get the lecture material. So this lecture was recorded. We're going to follow up by email and share the recording with you. You're welcome to watch that recording on YouTube as well. We have a YouTube channel where you will find a lot of free resources that you can go over. You can learn about the different programs. Like I said yesterday, we had the overview orientation for Introduction to Bioinformatics. If you're just getting started, you can go for the Introduction to Bioinformatics program. Today, we're talking about data science. And then tomorrow, we're going to be talking about transcriptomics with some specific focus on transcriptomic data analysis and interpretation. So thank you, everyone, for joining today. Again, look out for those emails with the recording for today, with all the links that we discussed, with all the different curricula. And thank you again for joining and for being here today. And we are looking forward to seeing all of you in the program itself. Okay, and Lane, did you want to say anything else?
No, just thanks everyone for coming to the orientation.